Well, thank you very much, Alan, for the introduction and also to the organisers for inviting me to this wonderful meeting. I first really interacted with David, I think, about 2009, when, coming from a very placental perspective, we wanted to try and bring together the fields of placentation and this emerging field of DOHAP. And so we organised a meeting in Cambridge where we brought together placental biologists and David and his colleagues, and this resulted in the publication of this book, The Placental and Developmental Programming. And today I'm going to try and summarise, I suppose, the contents of this book in 20 minutes or whatever. But I'm really just going to touch on 2 points. One is to consider the role of the placenta in developmental programming, just using one example of the materno-placental fetal dialogue in terms of transport, and then looking more towards the future to pick up on some of the challenges that were posed by David's later findings relating placental shape to adult disease, where I think we don't have any answers, but we can perhaps point some directions for future research. So the perspective we wanted to sort of bring to David was that there is this structure, the placenta, that is between the mother, her nutrition, and the fetus. And so we've got the maternal environmental cues here, and Tom has just alluded to some of those, in nutritional state. There'll also be her glucocorticoid levels, the oxygen availability, temperature and photoperiod can all interact through the placenta, and that will respond, as Tom has said, to some of these cues and adapt. But then that will determine the nutrient flow which actually passes through to the fetus, and then there will be interactions between the genotype of the fetus and that nutrient supply, which will determine its phenotype, which will be manifested then in the developmental programming of metabolism, endocrine disorders, cardiovascular disease that we're all familiar with. So the placenta, in our eyes, plays an absolutely crucial central role in this programming, sitting as it does between the mother and the fetus. And really there are a myriad of placental functions that could all be perturbed to affect fetal development. But I haven't really got time to deal with all of these. What I'm going to focus on is just really the exchange, because David's early thoughts were very much of nutrition, and it's a question of how do those nutrients get across the placenta to the fetus. So if we look, just to remind you of human placental structure, it's the hemochorial discoid placenta. We have the villus trees inside the placenta, which are bathed by the maternal blood coming from the spiral arteries. And this is just a cross-section of one of those villi, just to show you the relationship between the maternal blood here, the fetal blood in the capillaries, and at points on the villus surface, the barrier between the two is reduced to about 2 to 3 micrometers, 2 to 3 thousandths of a millimeter. So this is somewhat similar to the alveolus of a lung, and you might think in terms of diffusional exchange between the mother and the fetus occurring across this membrane. But in fact, there are several different mechanisms for materno-fetal exchange across that membrane, and this sort of cartoon summarizes those mechanisms. We can have simple diffusion, whereby nutrients cross the membrane, or they actually pass through what are thought to be fluid-filled pores. We can have transporter-mediated exchange, where we have transporter proteins on the apical and basal surfaces of the trophoblast, and that can be subdivided into sort of passive facilitated diffusion where there's no energy demand. Glucose is the classic example. Or we can have amino acid transport where there is a dependency on ATP. And then, as Tom has sort of mentioned, we can have endocytosis, exocytosis. So the placenta, even in just terms of exchange, is going to have the potential to adapt in many different ways to facilitate nutrient transport. The simple diffusion will be dependent really on the surface area and inversely proportional to the thickness of the membrane, governed by Fick equation. But we can also, in times of stress, increase perhaps the density of the transporter proteins that are inserted in the membrane. And just to show you an example of how these adaptations can occur, I think these adaptations sort of explain some of the variability in the data that you have in David's own data here, published between sort of placental weight along the x-axis here and birth weight on the y-axis. For any given placental weight, you can have a huge variability in terms of actual birth weight. And some of that may be due to adaptations within the placenta. Placental weight is a very crude measure, really, of placental transport efficiency. And so I think David's work really stimulated us to begin to look in more detail at some of these mechanisms. And of course, you move, as Tom says, to animal models. And this is quite a straightforward animal model where the mice have been fed just 80% of their normal diet. So this is an undernutrition model. In the mouse placenta, we have different zones. We have a labyrinth zone here, which is where most of the materno-fetal exchange takes place, and we have an endocrine zone around the periphery. And if you look at the proportion of these two zones, you see that it is quite different in the undernutrition mice compared to the control mice. So this placenta, for the same sort of weight and volume, can have very different transport capacities simply because it has more labyrinth zone at the expense of the endocrine zone. I mentioned transporters. We can actually do in the mouse transport assays, so you can measure transport across the placenta in vivo, and if you do that in these mice at day 16 or day 19, you see that amino acid transport is actually increased per gram of tissue in the undernourished pups. So why is that? Is that simply just this change in the labyrinth area? It also seems to be due to an increase in the expression of the genes encoding specific amino acid transporters. And so again, in the undernourished animals, we see increased expression on day 19 of this amino acid transporter here. So we see not both just structural changes, but also molecular changes in response to this undernutrition. The placenta is again sensing, as Tom has emphasized, the nutrient flow. Do those signals come from the mother? Is it picking up the nutrient supply coming from her blood, or is it actually responding to signals from the fetus saying, I need more amino acids? So that is a very interesting dialogue. And I think that's where we are today with this sort of concept, that we have here the nutrients coming from the mother, we have the placenta sitting in the middle and the fetus here. There are clearly signals coming from the mother to the placenta. The nutrient supply will impact on the fetal development, but equally we know now that there are fetal signals coming back towards the placenta that can alter this expression of these key transporter genes. So it is very much a two-way dialogue, and I think this makes it much more complex than just maternal nutrition alone. We need to think about how the placenta has developed and how it is functioning. And there are imprinted genes involved in here which are playing very important key roles in this balance between supply and demand. So this is a dialogue which has evolved really to try and ensure maximum development of the fetus, but also to preserve the well-being of the mother. So I think David's work on that nutrient flow has certainly stimulated research in this area. I want to just move on now, as time is short, just to consider his later papers relating placental shape to the incidence or the risk of adult disease in a variety of different conditions, from cancer to blood pressure, as we know. So this raises very important questions, I think, but quite difficult questions to answer. What are the determinants of human placental shape? And this is a question which remarkably I don't think has been addressed in recent years. So does it really matter whether the placenta is round or elliptical? Does it really matter how many cotyledons we see on the maternal surface of the placenta? And there are two, well, there's a fundamental question really I think that is raised is, does this affect placental function, or is it just a proxy marker of the implantation site and the intrauterine environment in which the placenta developed? In some of David's papers, he started to sort of talk as if the placenta had different functions on its long axis to its short axis. I find that a difficult concept to accept, really. I don't know of any evidence from placental biology that the placenta functions differently in different regions. For me, I think it is telling us much more about the placental intrauterine environment, which again obviously has impact on its function and the nutrient flow. So what determines placental shape? Well, initially in development we have villi, or the placenta, developing over the whole of the chorionic sac after its implantation into the wall of the uterus. At 6 weeks we can see here chorionic villi again over the whole of the sac. But then around the end of the first trimester the villi regress over the more superficial parts of the placental sac, and we end up with a more or less discoid placenta towards term. And a few years ago now, Eric Jonieu and I proposed the idea that this remodeling of the villi was induced during onset of the maternal circulation. So we know now that the placenta does not have much in the way of an arterial blood supply until the end of the first trimester. But more interestingly, when Eric looked with ultrasound at where the circulation starts, he found that it started more commonly in the periphery of the placenta than in the central regions. So diagrammatically, these are the spiral arteries, and we know that they are invaded by the trophoblast during early pregnancy, which is involved in the remodeling of those arteries. And that plugging of the arteries, these trophoblastic plugs, is more extensive in the central region here than in the peripheral region. So the circulation starts in the periphery. He gave us samples of placental tissues from the periphery and the central region. We looked at those. As you might expect, the oxygen tension is going to be higher here in the periphery than in the center, and sure enough we saw increased oxidative stress, increased apoptosis of the placental tissues, and decreased proliferation. So I think that this is one of the ways that is a powerful determinant of the placental shape, or in terms of remodeling the villi over the whole of the chorionic sac just down to a discoid placenta. And we went on to propose that abnormalities in terms of trophoblast invasion and the plugging of those arteries may determine exactly when that onset of circulation begins and dictate placental shape. So this is the normal case. We have the vessels plugged in the central region. We have higher levels of oxidative stress around the periphery. We get villus regression. We end up with a placental disc, the umbilical cord sort of more or less in the center. But if for some reason trophoblast invasion is limited in a particular area of the uterus, maybe there's low-grade infection, or there's many potential causes, onset of the circulation may be premature in this region, we get extensive, greater degree of regression of the placental villi, so that instead of getting a nice disc-like placenta, we end up with a rather raggy shape, and the umbilical cord is attached more to the periphery. If you get onset of the circulation throughout the whole placenta, we showed this with ultrasound again, you move towards the miscarriage situation. And that sort of ties with what you see at term, in that healthy placentas tend to be circular with the umbilical cord in the center. Placentas from cases of IUGR tend to be rather raggy looking with odd little pieces of villi that have been retained, whereas other villi have been regressed in the interstices. So that would fit with having poor trophoblast invasion and then poor subsequent blood supply to the placenta, leaving you with the IUGR situation. This, of course, is very difficult to prove, but there's recently been a paper from Kari Salafia's group where they looked at the site of insertion of the cord at the end of the first trimester and compared it to what happened you see at term, and they found a very strong correlation. So it does seem that placental, her conclusion is that placental shape is largely determined at the end of the first trimester. And we know now that IUGR placentas tend to be smaller at the end of the first trimester. Macrosomic placentas tend to be bigger at the end of the first trimester. Seems to be a correlation between placental size then and what happens at term. But of course the placenta is quite small at the end of the first trimester, so how does it grow? Well, Kathy Craven's group back in 2000 introduced the term of trophotopism, whereby the trophoblast migrated over the endometrial surface, and she believed that placental size increased through this sort of process, that the placenta recruited more spiral arteries as it grew. One problem with her work is that it was focused very much on the first trimester and examining specimens from 5 to 12 weeks. Now, I'm sure that this sort of thing happens in the first trimester, because that is how we get a larger conceptual sac and recruit spiral arteries. But it is difficult to equate this sort of view going beyond the first trimester. How do you recruit a spiral artery in the second trimester? How do you remodel it adequately so that it doesn't just spurt into the intervillous space? So I think this is valid for the first trimester. It certainly fits the pattern of trophoblast invasion you see across the placental bed, in that is greatest in the central region where the arteries are most remodeled, least in the periphery here where they've had less time to be remodeled, and that fits with the onset of the circulation that we saw. So then we've got to think about what happens after the end of the first trimester, and this is my own feeling of what happens, and I think this is an area we need to explore more, is how do the placenta and the uterus grow together, in that you could, if the opposite sort of view would be to say that it is uterine expansion that is taking place. The placental footprint on the uterus remains the same, but in fact, as they both co-enlarge, then placental diameter increases from about 5 centimeters at the end of the first trimester to something like 22 at term. So you're talking about a 4 centimeter of a fourfold increase in diameter. If you go with this sort of view, then you would expect the spiral arteries to move apart as placenta, as gestation advances. So in the non-pregnant uterus, and it's very difficult to get data, just simple anatomical data on the human uterus these days, it seems that the spiral arteries are about 2 to 3 millimeters apart. Each spiral artery opens into a lobule, or rather a lobule is formed over the opening of a spiral artery. And these lobules in the mature placenta are about 1 to 2 centimeters in diameter. So if you go on that sort of argument, you're saying that the spiral arteries have moved from something like 2 to 3 millimeters apart in the non-pregnant state to something like 1.5 centimeters in the term. And again, that's a sort of 5-fold increase in expansion. But I think this is an area that we need to sort of explore much more in relation to David's work. What you see is that lobules are not of the same diameter throughout the placenta. The ones in the central region tend to be larger than those in the periphery. Now that may be because those peripheral vessels haven't been remodeled properly, or it may be that the periphery of the placenta has not expanded or as much as the central region. So the question rises as to how evenly does the uterus expand during pregnancy? Again, very difficult to actually find any quantitative data, but this is taken from Gray's anatomy. I don't know how accurate this is. I think this is an area where we need modern imaging to sort of tell us more about the expansion of the placenta and the expansion of the uterus. Because if you look at this, we've certainly got a sort of expansion of the fundic region here at this stage compared to here. So if your placenta was implanted in this region here, it may well stretch in a different way to one that is implanted just at the top of the fundus. So I think these are very interesting questions that we should perhaps now address in the light of David's work. Does it tell us about placental function? Well, there are some very basic findings that we don't know. What is the density of spiral arteries and uterine glands which are going to supply the early placenta throughout the uterus? I can't find any data in the literature which has sort of looked at regional variations in the lateral wall of the uterus, the anterior, posterior wall, or the fundic region. So I think that's a big area that we perhaps should pay attention to. We speculated that differences in trophoblast invasion and onset of the blood flow could determine placental shape. I think we need to think about local variations in maternal immune cells or low-grade inflammation influencing trophoblast invasion. And is development of the uterus itself vulnerable to programming? Are the spiral arteries and the glands the same in a girl that was born IUGR? Certainly the size of the uterus seems to be smaller, but we don't know about its internal structure. So I think these are some of the questions that, of David's legacy for the uterus. Similarly, we can think of any number of questions for the placenta. What are the critical periods? He talked about the number of cotyledons, which are determined really by these septa projecting into the placenta. We have no idea about what the formation of those septa is, what their physiological significance is. He talked in terms of placental thickness. Again, we have no knowledge really of what determines placental thickness, and we don't really know about the branching of the arteries over the chorionic plate. So I remember David submitting manuscripts to Placenta. They would go out for refereeing, and people would say, well, these are very interesting observations, but of course there's no mechanism. The reviewers were really then quite harsh about the paper. And then I would send them back to David, and David would ring me up and say, well, of course there's no mechanism, because these are observations. You, the placental biologists, should think about these sort of questions now and come up with the biology that is underlying these. And I think that's exactly what Tom was reflecting, really, that David posed a number of questions for us, and it is now for us to tease out the underlying biology. So I think David's research provided a huge boost for the placenta, and really a raison d'etre, if you like, for placental research. I think virtually all grant applications that go in now on placental research will be prefaced by David's findings. I think we are beginning to tease out some of the details of the materno-placental fetal dialogue that is determining resource allocations. It's not just placental weight, that there are subtle changes that need to be borne in mind. I think placental shape and the trajectories for placental growth are probably largely determined by events at the end of the first trimester, and for me the first trimester is absolutely critical for placental and fetal development. But there is still a huge lack of knowledge regarding the basic anatomy of the uterus and the relationship between the growth of the uterus and growth of the placenta during pregnancy. And I think that's sort of David's legacy to us. So thank you very much for your attention.